First, okay. Good afternoon, everyone. I'm Renee Prince from CenGen, a plant genetics company based in Worcester in the Brede River Valley. And it's my privilege to officially welcome you to the Veldkos Field Food DNA project, a milestone for life sciences education in South Africa. The meeting link was shared freely with many people, and since I'm not sure who is attending, I do not want to single anyone out. But however, at this point, some thank yous are in order. I want to thank Lucy Olivier, Senior Education Specialist for Life Sciences in the Cape Winelands Education District, for her incredible support to put this project together. The participants will introduce themselves and the schools they represent later, but I want to say a huge thank you to all 6 schools headmasters and life sciences management teams who welcomed our idea with open arms. Dealing with such enthusiasm in the education community is a very positive experience. We have a very diverse audience today, and in addition to the participants and the teachers, we also have representatives from different governmental departments, the media, funders, and colleagues. And therefore, I would like to go through the layout of this meeting so that you can identify a time that you might want to leave. We won't be offended and appreciate your attendance at the start of this meeting. I will start off with a background to how this project started, and we are quite a lot of people involved to make this happen. So I would like to introduce you to key team members and also funders. Then Elizabeth will set the scene in terms of theory to introduce these learners to plant genes and genomes, since they are grade 11 learners and haven't had a lot of exposure to DNA and genetics yet. Immediately following her talk, we will allow questions on the theory part. Then we have a short break of 5 minutes. Then Helmine and Larisha will take you further with the practical work and the data analysis. So that will be quite a lot of detail discussing with the learners how their week in March will look like here at CenGen. I will just touch on a few logistic things very briefly, and then will have ample time for questions where we will open the floor. So you are also welcome if you want to join and leave for a while and come back. And then Elizabeth will conclude the meeting with a nice activity in Mentimeter. Yeah, so let's get going. So this training initiative has 2 important goals. Firstly, we would like to introduce potential future scientists to the exciting field of genomics. And secondly, generate useful genomics data for an important plant species that has been scientifically neglected thus far. So the diplomics made us aware of the UK daffodil DNA project, which originated after an investigation revealed that learners need to be exposed to real science to consider a career in science. And in the daffodil DNA project, they performed advanced genomic experiments on different daffodils at their schools under the guidance of their teachers. Now, in this South African initiative, we mirror some aspects of this daffodil project, but we are very cognizant of the fact that our schools are already under severe pressure. And therefore, the project's practical aspects will rather be performed by the learners at CenGen's newly established training lab, which was made possible through diplomics. But at our core, we have scientists, and we also could not pass up on the opportunity to make a valuable contribution to science in South Africa. Therefore, we chose, in collaboration with the famous botanist, Dr. Ernst van Jaarsveld, a group of plant species that have received very little attention in our country. These learners are going to help us establish the first genomic resources for a Veldkos or field food plant species. And these plants are what we call the so-called forgotten food species. They may become an important source of food in the current changing climatic conditions. We wanted to identify learners that will seriously consider a career in life and or computer sciences, and therefore the decision was made to give 2 learners of each of the top 6 performing Cape Winelands Education District schools in the 2022 NSCC Life Sciences exam an opportunity. These learners also made a serious commitment by officially committing 100 hours of their free time to participate in this project. For that, we are very grateful. This whole training initiative has been designed to expose the learners to the real world of science, which includes the grant writing application process. And therefore, I can report today that we were partly successful in securing funding for the project and that we had to do things like change the final media event I spoke to you about to an online event. We will also only know by the end of April to what extent our UK partners can be involved when the outcome of our application to the British High Commission will become known. We hope by the end of this year that you as participants will have an excellent understanding of the exciting world of science, what it entails, and we want to encourage you to share what you are learning as you go along with your fellow classmates. This would not have been possible without our main funder, Genomics, an initiative of the Department of Science and Innovation, and therefore I would like to hand over to the Program Manager of Genomics, Dr. Tim Newman, to say a few words. Thank you, Tim.
Thank you, Renee, for that introduction. I'm assuming everybody can hear me clearly.
Yes.
Okay, great. So thanks everyone for being here. There's many more people in the audience than I expected. I've just put together a few slides to give the learners an idea of who we are and what we do. I'm not used to speaking to high school learners, so hopefully I don't overshoot some of the topics I'm going to go through. But let's go ahead and get started. Renee, the next slide, please. So Diplomics is an acronym. So it's used to describe what we call a distributed platform in omics research. I'll get back to omics just now, but this is really it's a government funded program to support the growth and development of high-end laboratories that do genomics and proteomics research along with bioinformatics, which is the ability to analyze that data in the country. Next slide, please. So omics is a fairly complicated field. The word omics itself is actually a Greek suffix, which means all. So it's been attached to the end of a lot of traditional areas of study like genetics, transcripts, proteomics, and so forth. And it just means that the study of everything in a cell, either at the gene level or at the transcript level or proteins. So omics, I like to break down into these categories. So you've got the biological systems, which are at the core of what we study. And those systems have their own sciences, which I've just mentioned. So genomics looks at all of the genes in a cell or an organism. The transcriptomics would be the DNA, which is a copy of, the RNA, sorry, which is a copy of your DNA. And that's like a recipe for building proteins. And you'll be hearing more about that, I assume, a little bit later today. But then we have the methods in omics, which is how do you go about doing these sciences? And so under here, I've got the two sort of key broad areas we normally talk about. So genomics, including transcriptomics, which uses sequencing or microarrays. You'll be learning about sequencing a little bit later, I hope. And then when we study proteins, we use instruments called mass spectrometers. I heard somebody describe them as very, very sophisticated scales. They weigh proteins. And then finally, we've got the technology we use to study omics. And these are these instruments. You don't need to know what they are, but they're typically very expensive, very sophisticated, and often take a lot of training to learn how to use. And then the core, or actually the underlying science that pulls all this together, is the bioinformatics. Next slide, please. So the distributed part in the word diplomics is the fact that we are actually a network of partner labs throughout the country, CenGen being one of them. And these networks are all pre-existing labs. Most of them sit within a university, but some of them sit in government institutions, or in one case where I'm sitting today, a private company that offers omics services to everyone. And we have a number of labs, most of them genomics based, and we have a lot of people that either work directly or are affiliated with these laboratories. So we have quite an extensive reach. Next slide, please. So the two main objectives of our program is to build a world-class omics infrastructure. I won't go into that too much. And then equally important, if not more so, is to enable access for students and researchers to that infrastructure. The infrastructure just means the collection of the instruments, the buildings in which they sit, and everything that's needed in order to have a successful and functioning laboratory. So we buy instruments, we place them in labs, we help ensure that those machines are well maintained, that the technicians that work on them are also trained and know what they're doing. And all of this is to ensure that all of that expensive instrumentation and hardware is up and available and ready to do science. And then on the enabling side, we do lots of efforts in training, which is why we are sponsoring this event or contributing towards it, because training is essential. If we don't have people coming through the system, especially high school learners who are interested in pursuing these sciences, then all of this infrastructure doesn't matter. So the training, skills development, and so forth is quite critical to what we do. And part of this is ensuring that people throughout the country at least have the chance or the exposure to understand what omics is and hopefully get access to it through training or through hands-on laboratory experience. Next slide, please. So the fun part of what we get to do is buy these fancy instruments. We spend a lot of time understanding what new technologies are on the horizon. And we are fortunate to also have been able to acquire instruments that in many cases are the first ones in Africa or in South Africa. And at the top is just some examples of sequencing instruments. These happen to be from one supplier that we work with. There are many more. And then on the bottom are just some examples of the proteomics side. And on both sides, we've been able to bring in very cutting edge technology, which is good for you to know because it means you can do research here that people anywhere else in the world are conducting. Next slide, please.
Oops, sorry.
Yeah, back one. Yeah. So part of our job is also to make smart decisions about instruments. I said they're expensive, and I'm going to give you an example. On the left is a sequencer, a genome sequencing machine. It costs 25 million rands, and it weighs 800 kilos, and it's the size of a refrigerator. And at one point along the way, we decided, well, that's actually more than we need. And with the same amount of money, we could buy 137 small sequencing devices. I believe you'll be seeing some of these during your course. I hope I'm right, Renee. And these are mobile, self-contained. You can take them almost anywhere and sequence genomes, not quite at the same level as the machine on the left. But for us, this is a much more effective way of spending our money. That's just one example. Next slide, please. I mentioned that training is important. We call that human capital development. We have done some other high school outreach programs where we bring in students who get to get a little bit of lecture like you will get and also have the opportunity to extract some DNA. It's very successful and very popular, and we love doing these kinds of things. Next slide, please. And then the next level up is we provide training all over the country. Sometimes we hire companies to do that for us, but increasingly we are funding our own partners like CenGen to provide the training themselves. And this is very exciting because the more that we train people, the more they gain the ability to train others. So it has a really nice pay it forward type of effect. Next slide, please. So finally, if you want to know more about diplomics or you want to know more about omics, we have a website, diplomics.org.za. I don't know if you can see it there. I'm sure you'll get that information through the course. Next slide, please. On that site, we publish a lot of videos. We do videos that profile our labs. It tells you a little bit about what they do and why omics sciences are very valuable and important for the country. Next slide, please. And also what important for us is to profile the individuals in science. We usually celebrate the Women in Science Week by interviewing women scientists, so you get to know more about what they're doing and why they're excited about their research. And we also do videos on some of the technologies that we use. Next slide, please. Yep, that's it. So I hope that was of use and of interest to you. Thank you very much. And I don't know if there's a Q&A session, but you can always send me questions later if you like. Thank you, Renee, and to the rest of the team.
Thanks a lot, Tim. You made a complex issue very simple. Thanks a lot for that.
You're welcome.
We also want to thank and acknowledge the John Innes Centre in the UK as a funder. The JIC is a very exciting and vibrant place where scientists from all over the world converge to excel in plant sciences and microbiology. And we are very grateful for the time and funds that their informatics team, under the leadership of Dr. Burkhard Steuernagel, will invest in this project. This collaboration will also provide some insight for the learners as to what it will take to be internationally competitive. And here are the CenGen team members who will work alongside you during this project. You will see that in a research or a scientific team, there are usually various team members, each with specific roles that complement each other. And as you can see, we all had very different training paths, but no one's role is more important than the other. We only indicate our degrees in brackets to give you an idea of the diversity in terms of education, but we don't use titles here at CenGen. And Ernst van Jaarsveld from Babylon Store and Farms and the University of Western Cape is also a key member of the team and the supplier of the plant material you will be working on. But research teams often consist of team members from across various institutions that contribute specific skills to the project team. And we are very fortunate to have access to the incredible skills and training experience of Burkhard Steuernagel's team, who will most likely join us in person for the training in week 2. Roland already played a huge role to help us establish a DNA extraction protocol that works well for plants and which you will be using here during your visit. And Burkhard and Martin will introduce you to the incredible world of data science or bioinformatics. The support from the education department is incredible. I already mentioned Lucy, who was instrumental in realizing this initiative, but we are also grateful for Jo Marie Schultz, a teacher not affiliated to any of the 6 schools involved, who will help us to take care of the learners while visiting our lab here in Worcester. Thank you, Jo Marie. And now we get to the most important people of this initiative. I hope the 3 learners who struggle to get in are joined the meeting. But it's you, the participants, what this meeting is about today. So I want to ask you to switch all your cameras and microphones on. You will be the scientific team and executors of the project. And they will now introduce themselves. Please, you don't have to rush. State your name, say from which school you are. And remember, there's a lot of people here from all over the country. They don't necessarily know where your school is based. So please tell us also in which town is your school based. Thank you. Lizelle, you can start at the top.
Hello, everyone. My name is Lizelle Abbott, and I am from La Rochelle Girls High School in Paarl.
Hello everyone, my name is Isabella Douglas. I am from Robertson and I go to Robertson High School.
Yes, hello, Yella. My mic isn't working. Hello, Yella.
It's better,
My
yeah.
name is Joshua Duplessis, and I'm from Paul Rus in Stellenbosch.
Hi everyone, I'm Marisa Tue and I'm from Paul Gulzo in Paul.
Did Caitlin manage to get in?
Unfortunately, Caitlin is still struggling, so she said she'll watch the recording.
Okay, Daniela, please proceed. Miss Daniela here?
Daniela was online to join by phone, but I don't see her online at the moment.
Okay, Dosh. I think your name is spelled incorrectly. Apologies for that.
No worries. Hi, my name's Dosh. I go to Paul Rus in Stellenbosch.
Okay.
Hi, I'm sorry my camera does not want to work, but I am Caitlin Henderson and I go to La Rochelle in Paul.
Xenia managed to join?
Hi everyone, my name is Zania and I go to Renish in Stellenbosch. Yeah.
Hi everyone, I'm Heidi and I go to Gulzo in
Hi everyone, I'm Jody and I go to Robertson High School in Robertson.
Hi everyone, I'm Anouk and I go to Bloemhof High School in Stellenbosch.
Petrus?
I am Petrus. I am from Worcester and I go to RTS Thursday.
Okay, great. So thank you everyone. Nice to meet you. And you can turn off your cameras and microphones now. Thank you. So just a layout of the year's program. It's a 1-year project. And the two major events will be week 1 and 2. In week 1, they will come to Worcester to do all the practical work. And in week 2, they will come and analyze the data. So they will introduce to the wonderful world of bioinformatics and working with coding and on computers. So the Center for High Performance Computing, what we call the CHPC, and the Data Intensive Research Infrastructure of South Africa, the RISA, are two of the 3 primary pillars of the National Cyber Infrastructure Intervention supported by the DSI. And we use the CHPC for large-scale data processing and the RISA for long-term data storage and management. And you will see the extent and magnitude of the type of data that you will be working with and why these wonderful resources, why it's so wonderful to have access to these resources. And this is all made possible through diplomics. We will therefore be in between these and post these visits, we will host another 7 online in-depth walkthrough with the participants only and their teachers. And that will be to specifically discuss the project as we go along, results and future work. And those dates we will try to finalize when you are visiting us in March so that everyone knows well in advance the dates. Very early in December, just after your important grade 11 exam, and before the teachers go off to mark their NSC papers, we will host an event where you as a scientific team will present your data. So Elizabeth will now set the scene to establish a genetic basis for you. Enjoy.
Okay, good afternoon, everybody. My name is Elizabeth, like Renee said, and unfortunately, I'm not in school anymore. But I live in Paul and I work here in Worcester at CenGen, and I'm going to tell you a bit more about DNA, and we're going to talk a bit about concepts that you will be hearing once you are here. And hopefully that will help you not to be too lost when you hear about these things as we talk about it when you visit us. So we're going to jump right in. Each research project always has an objective. In other words, what you want to do. So the objective of this project, as you all know very well by now, is we're going to sequence the genome of the groot noem Veldkos species. But that's all nice and well, but what exactly is a genome? You might be or are hopefully familiar with plant cells by now. Plant cells are different from animal cells in a few ways. The major difference being that they have a cell wall, which animal cells don't have. And then they have extra organelles like chloroplasts, which are responsible for photosynthesis and causing the plants to be green. But this might make it a bit more tricky to work with plant cells. We'll get to that a bit later. Inside of the plant cell, or any cell for that matter, you have the nucleus. It has a membrane around it, and then you have the nucleolus inside of that. And inside of that, you have the genetic material. In other words, whatever you inherit from your parents. And it's the same for plants. Plants also have parents. They inherit the traits, the way they look, the way they function from the two plants that were crossed to make them. And this genetic material is visible during certain stages of the cell when the cell is busy dividing as what we call chromosomes. So each species has a specific number of chromosomes. It's very specific to that species. For instance, humans have 2 sets of 23 chromosomes. An apple has 2 sets of 17 chromosomes. And then wheat, what you would not expect, because wheat is not a very impressive plant, but wheat has 6 sets of 7 chromosomes. So it's got quite a lot of chromosomes, very complex. And we don't know how many chromosomes the groot noem species has. So if you unravel chromosomes, if you pull the chromosomes apart, then you will see DNA. So usually when the cell is not busy dividing, when it's just doing its usual thing, then the genetic material is like a jumbled up molecule, and it's called DNA. DNA is a very long strand, a very long molecule, and parts of the DNA molecule in certain sections are what we call genes. You might have heard of genes before. That's a bit more a familiar concept. That is what causes you to look the way you do. That is what causes plants to function like they do, animals as well. So if we look a bit more closely to DNA and DNA structure, you will see DNA looks like what they call a double helix. Now, if you imagine one of those spiral staircases, it's more or less like that. But then when you untwist the DNA molecule, so you basically pull it straight, then you see, or you'll see that it looks basically like a ladder. And the 2 sides of the ladder are made up out of sugars and phosphates. And then the inside, so the steps that you step on, those are nitrogenous bases. And you've got 4 different ones. It's very important to remember that all of this is not visible under a normal microscope. It is just too small to see. But we have ways of looking at it in the lab. So if you look at these bases, there are only 4, like I said. So you've got an A, a T, a C, and a G. And they are arranged in a specific order on the DNA molecule. And that is very important. What's also important is to know that they always pair up. So the A stands for adenine, and adenine is always paired up with thymine. And then you've got the C for cytosine, and the G for guanine, and those 2 are always paired up. So you will never see a thymine paired up with a cytosine in normal DNA molecule. That is not the way it works. So they are always paired up. And yeah, then you've got these 2 backbones that connect them. So if you then pull it apart even a bit more, you will see that, or you can imagine that if you have the sequence of this one strand, you don't need to see the other one. You can actually figure it out because you know now that A always goes with T and G always goes with C. And it doesn't matter if the C is on that side and the G is on the other side, they still go together. The way we read DNA is it's always from the 5 to the 3 prime end. That's what we call it. And that is what we call the forward strand. And then the opposite direction, the 3 to the 5 prime, that is the reverse strand. And the reason why it's called 5 and 3, I know that's a bit weird. It doesn't make sense. Why not just call it the front and the back? The 5 and the 3 refer to the type of molecules that sit at the end of the DNA of this molecule. But that's not important for now. It's just important to remember that DNA is always read in the direction 5 to 3. So this strand, the reverse one, is now in the incorrect direction because it reads 3 to 5. So to get that into 5 to 3, you have to swap it around. And then you always have to make sure that the A still goes with the T, and that what we do then is we create the reverse complement strand. Okay, so now you can ask me, we only have 4 of these bases. It's not the whole alphabet, it's only 4. And how can I say that these 4 bases of this genetic material molecule of the DNA, how on earth is that responsible for all the functions in the cell and all the things that you see in the organism, like smell, things like sound, how plants defend themselves, how people differ from each other, how things grow big and small, how things function. How on earth are only 4 molecules responsible for all of that? It doesn't seem like that's right, but it is. And now we get to what we call the central dogma of molecular biology. Big word, but don't worry, we will explain to you what it means. And basically, if you break it down, it means that DNA, the message from the DNA is transferred to RNA, and from there it's transferred to proteins. And proteins are the workhorses in the cells. They are what's responsible for everything that's happening and how things look and how things function. So to explain to you how this process works, remember you've got the cell. Inside the cell, in the nucleus, you've got the DNA. The DNA looks like this. You know by now it has 2 strands with the bases that are connected to each other. Then the DNA message or the code, in other words, the order of these bases, that is transcribed. So transcribed just means you write it over. So it's basically written over into mRNA, messenger RNA. Now, RNA is very similar to DNA, except probably the biggest difference is it only has 1 strand. It doesn't have 2. And in RNA, the T is replaced by a U, but it's still the same thing. So if you look closely at the 5 to 3 strand here, you'll just see TU, the A is the same, the C is the same, the G's are the same. So it's literally just written over into RNA. Then from RNA, the message is translated, in other words, written into a new language. And now it gets translated into proteins. Proteins are made up out of amino acids. So now you move from bases in DNA and RNA to amino acids. And how it works is in RNA, the bases are basically grouped together in groups of 3. And those are called codons. And one codon translates into a specific amino acid. And all these amino acids together are what are making the protein. And there's a specific code for it. This doesn't just happen randomly that you have a serine here and a glycine here. This code is translated very meticulously, and we can look at it in this way. So if you read from 5 to 3 again, let's say you start with an A, and then the second of the 3 bases is an A, and the third one is a G, then it makes a lysine. So that's the amino acid that gets made. If the A changes, or if one of your letters changes, then your protein will also change. So let's say you had an AAG here. Let's say now you have a G, and then an A, and then the G again, you have a glutamine. So these things are responsible for the changes. And if you have a random change in your plant or in your cell, or maybe it's human, then your protein might change because now you've got a different amino acid, and then the function of the protein might change altogether. So remember when I said initially that a specific piece of the DNA at a specific position of the genome, that is called a gene. So we're going to look a bit at genes now, next up. So this is an example of a plant gene. Genes usually have quite intricate names. This one is actually quite simple. It's called the PH1 gene in the petunia. You might have seen petunias in a garden somewhere before. And the very fancy way of saying what the gene does is it codes for a protein that boosts the activity of a pump in the tonoplast of petal cells. What does that mean? So remember, inside a plant, you have a vacuole inside the plant cell. The vacuole has a membrane. It's called the tonoplast. You might have learned this last year. So if you think of the cell, on this side of the tonoplast, you have the cytoplasm, so the rest of the cell. And on this side, you have the inside of the vacuole. This protein, the PH1 gene, makes a protein, and this protein sits on the membrane. And it literally just pumps hydrogen molecules from the cytoplasm into the vacuole. And as it does that, the color of the petunia changes and it becomes purple because the pH lowers inside of the vacuole. So it's actually not that hard, and it's quite nifty the way these things are designed. If you have a change, so let's say one of those bases changed, now you have a new codon, then you have a new amino acid, your protein changes. If that happens, then the petunia is not purple, it might be blue, if that change is a significant change. So that is the way these bases, these 4 bases, influence how a protein functions in the end, how the gene, how the plant looks. It's all connected. So if we look at numbers, remember I said the gene is a piece of the DNA. It consists out of those 4 bases. Let's talk about how big are genes. So the petunia gene that we just talked about is about 7,300 bases. That's one gene. So let's say you write them all on a page, all these letters on a page, the A, C's, D's, and G's. You can fit, if it looks like this, you can fit 7,120 bases onto one page. So that's a gene. Now, if you go to a simple organism like this bacteria, it lives down in the ocean, its entire genome, in other words, all its DNA, is 3.6 megabases. In other words, 3.6 million bases. If you type that out, like you typed out the gene, it will take up an entire pack of printing paper. So 500 pages. Let's go bigger. We've got the human. The human has 3 gigabases contains 3 gigabases, the human genome. That is 3 and 9 zeros, 3 billion bases. If you type that out on pages and you put the pages next to, like head to toe next to each other, you can fill the road from Worcester to Hermanus with paper. So it's a lot. It's over 4,000 pieces of paper, and that's all the bases, the DNA bases of the human genome. We can go even bigger. Remember I said in the beginning or early on that wheat has a very big genome. Wheat, you wouldn't expect it because it's just a plant. How can it be bigger than a human? But the wheat genome, 15 billion bases. That would get you up to Koolsberg if you start putting papers down from Worcester. It's 600 kilometers of paper just to type out the entire genome of wheat. And this is really, really, really big. So we don't know what the size of the groot genome is at this point. You will not be determining the size, you will just be determining the order, the sequence of these bases, and that's really important to know. So how do we read this? I mean, there must be some way of reading this, and that's what you want to do, is you want to read this piece of paper or these books, basically. We need to get out the DNA first. I mean, the DNA, you don't want the rest of the cell, you only want the DNA. We regard the rest of the cell basically as gunk when you work with DNA. But remember, when you extract DNA, a piece of leaf doesn't just have one cell. So you start with a piece of leaf when you do a DNA extraction, but you have multiple cells, thousands of cells in that piece of leaf. So you're not going to extract only one copy of this genome. You're going to have multiple copies because you're extracting from multiple cells. And that's important to remember. You won't only have one piece of DNA, you'll have lots. And then we have these instruments, and Tim mentioned the sizes of instruments. We don't have a big one like that. We're going to use a much smaller instrument, but just as effective. It's called the Oxford Nanopore P2 solo sequencing instrument. A lot of words. Helmine will tell you a bit later how it works. She will show you, Larisha will show you a little video. But for now, it's important just to know that this little instrument is what is going to be doing the reading of this book, of the sequence. So now I said you have different sizes or different pieces of DNA that you've extracted. Now you have to piece them all back together because you don't, it's like building a puzzle. You need to figure out how they fit together in which order, because remember the order of the DNA is very important. If you have small pieces, so let's say from your DNA extraction, your pieces are very small that you get, and your instrument cannot read very long pieces, so it reads little small pieces of your sequence at a time. You can start off. So let's say you've got this jumble of sequences. Now the sequencer is trying to piece them together. It's trying to figure out how they fit. So it's basically looking for overlaps. So if you have this TAC, it can go in the middle of the long one piece, but it can also go to the front, and you're not quite sure where it fits. The sequencer has to figure that out. And then you can continue building your sequence. But now, at some point, you're going to realize that the pieces you have left don't fit in with the piece that you've built already. You start a new piece, and you think, okay, later it will be, something will come along that will connect these two again. So you continue building, and then the second base that we put in here, this little piece, it's only one that overlaps. So how confident can you be that that's actually the right place for it? Does it really have to go there? Does it go with a third batch of pieces altogether that you don't know yet? But let's say you've got this last piece. What you can also do, what can happen is this piece can be swapped around. So remember the reverse complement. So you can literally have the other side. And remember, if you had an A, a C, and a G, now you have to read it from the back, and you have to translate it. So now you've got the G goes with the C goes with G, and the A now goes with the T. Now you've got a new sequence, and that sequence now moves. So now it fits in a different place. And then in this case, we've got these two AAs at the bottom, and they don't fit anywhere at all. So now what do we do? And that's where the Oxford Nanopore technology is actually very nice, because it's able to read longer sentences, longer pieces, bigger pieces of DNA. And if you then start matching them up, then you're much more sure of what you're doing. You've got a lot more confidence because now the pieces actually fit together nicely, and you know, okay, there's a lot of them in the same place and they all work nicely together. So you're much more confident about the puzzle that you've just built and that you have the correct sequence that you're working with. And the science behind this is what we call bioinformatics. So you've done the wet lab, we call it the wet lab, the actual DNA extraction and setting up the sequencing experiment and doing the lab work for that. But this analysis, this building the puzzle and adding the pieces back together, that is what we call bioinformatics or data science. Some places where you study it, it might be called data science. And this is a very exciting field. Larisha will tell you a bit more about this. And this is computer work. So now you move from the lab in more to computers, and now you have programs, and you can do some coding that will help you, or that will eventually build your puzzle. So I just want to recap. Right at the end, remember, cells have DNA inside their nucleuses. That is what is inherited from the parent to the offspring, and that is what makes us all unique. That is what makes plants function, what makes hearts pump and bacteria grow. DNA is responsible for how that happens because it translates into proteins. Remember, DNA is in the nucleus of the cell, and in a specific piece of a plant that you will have many, many, many cells. And together the DNA is called the genome. And this is now to latch onto Tim's talk. If you study the genome, the genetics, you are busy with genomics. So that's my talk. You will be able to ask some questions now if you want. Don't be shy. You're also welcome to ask them in Afrikaans, and I can answer them in English for everyone else. So over to you, Renee.
Thank you, Elizabeth. I just want to say that Caitlin Aronson managed to join us and we haven't met her. Caitlin, can you switch on your camera maybe and just say from which school you are and in which town? Francois, will she be able to do that? I can't see her.
Hello.
Hi, okay, great. Welcome, Caitlin.
Thank you. I'm Caitlin Aronson and I'm from Rhenish in Stellenbosch.
Okay, welcome. We'll catch up. Don't worry about that. And just before I had a request, leave your camera on. I want to ask everyone attending if they don't mind to switch on their cameras. The Plomix Melikazi requested us, they want to take a group picture. And I think it's a good time now, just before the questions. So everyone, can you please switch on your cameras? Melikazi.
Renee, you're on mute. Huh?
Sorry. Will Melikazi do this? Because I don't know how to do this. Take a picture, Tim.
I guess we can do a screen grab. I'm only seeing 2, 3, 6, 8 photo,
No, we still need 26
cameras on.
others to join.
Yeah.
How will we do that?
Unfortunately.
Would you like
Well, I'll
put our cameras on as well, or just the students?
Everyone, they asked the group. Everyone, please switch on your camera if you don't mind being in the picture. Then is Melikazi close to you? I don't know, where is she? Jane, are you in the meeting still?
Maybe that answer.
I don't know where she is now. Okay. I think let's carry on. We can't. Let's carry on and then we'll ask again at the end of the meeting. Apologies, guys. So you're welcome now to ask any questions about this theory section. And please just you don't have to remember everything. We will repeat a lot of this during the workshop. But if you have any questions, you're most welcome. Okay, no questions. So it's now 15:53. Let's start again at 1600 hours. Please don't leave the meeting. If you want to have a quick break, we'll start the presentations again at 4 o'clock. Thanks a lot.
Welcome back, everybody. So I am Almin Barkes and Roda, and I'm going to tell you more about what will be happening during your first practical week here at CenGen. Sorry.
Yeah, is there someone with a question? No? Okay, Almin, continue.
So I'm just struggling to get my mouse to work. So okay. So what is the aim of our practical week? So we want to get groot bloem leaves, or we want to get from the groot bloem leaves or the plant cells to an actual stretch of DNA. So before we can use that, or before we get to that, before we get to how we are going to do this, there are some important things that you need to know regarding your practical week here in the lab. So you've probably heard of personal protective equipment, PPE, especially during COVID, the word PPE. So working in the lab, it's very important to wear personal protective equipment. So this includes a lab coat and gloves. This is for you to you don't want to spill any of the reagents on your skin or on your clothes, but you also don't want to want any of your own DNA to contaminate your samples. So during the first day of the practical week, you will work with liquid nitrogen. And liquid nitrogen is the temperature of -80 degrees, and it often boils and splashes. So it's very important to wear additional PPE. So you'll also wear thermal gloves and safety goggles when working with liquid nitrogen for your own protection. And we'll also work the first day with chloroform. So chloroform is a colorless liquid that quickly evaporates into gas and is also toxic. And for these steps, you will be wearing a mask. A big part of your practical week will be pipetting. And when you hear the word pipetting, you're probably thinking of this. But here at CenGen, you are going to work with micropipettes. So this is how they look like for those of you that haven't ever seen a micropipette before. The biggest volume that you will work with will be 1 mol. So think of the smallest volume that you can measure with baking measuring spoons. And then you'll also work with volumes as small as thousands of a milliliter, which is 1 microliter. So next up, I'm going to show you a video just of how to use micropipettes. The lady's voice is a bit squeaky, but it gets better towards the middle of the video. So just bear with us.
Hi there, I'm Jamie Dyer. This is a micropipette. It's the most important tool in the molecular biology lab. I randomly picked this issue of the scientific journal Nature, and all of these articles did at least some part of their research using micropipettes. A micropipette transfers tiny volumes of liquid. It does this by drawing up the liquid like this. Of course, in the lab, we don't transfer liquids using straws. We use pipettes. Pipettes transfer milliliters of water, while micropipettes transfer microliters of water. Micropipettes come in a few different sizes for the same reason we use different scales to weigh different types of animals. A laboratory scale is sensitive enough to weigh a mouse, but not an elephant. Similarly, this micropipette is sensitive enough to pipette 5.5 microliters, but it doesn't have the capacity to pipette 500 microliters. These are the most common ranges for micropipettes, though there are other ones as well. We name them according to their range's upper limit. This is a P20, P200 and a P1000. If I need to transfer 800 microliters, I'll use a P1000. But if I need to transfer 30 microliters, I'll use a P200. You can tell what the range of a micropipette is because it's written on it somewhere, usually on the plunger, but not always. When you use a pipette, you make sure to draw up the right volume of liquid and looking at where the liquid is on the pipette. But micropipettes transfer such a tiny volume that you can't be accurate just by looking. So for micropipettes, you set the volume and the micropipette will draw up exactly that much. To set the volume on a micropipette, you turn this dial here, and these numbers show you the volume that it's set to. The unit is always microliters. So this P1000 is set to 650 microliters, and this P200 is set to 85 microliters. But be careful, because for micropipettes with a range of 20 microliters or less, the volume display includes an extra number. It's usually red, which represents decimals. This is 12.5 microliters, not 125 microliters. Never set the volume above or below a micropipette's range. It will break it, and these things are expensive. If I didn't have a straw, I could transfer the liquid like this. But now this is contaminated with my spit. The opening of a micropipette is a little bit like a mouth, and it's usually pretty dirty. And it's not designed to hold liquid anyway. Never put a micropipette directly into liquid. Just like I used a straw, a micropipette needs something clean to hold the liquid. That's where this comes in. It's called a tip. The tip is clean and it holds all the liquid. Different micropipettes need different sized tips. There are a couple of ways to tell which tips fit on which micropipettes. Sometimes the tip box says what size tips they're for, but not always. Often the color of the plunger is the same as the color of the tip, but not always. You'll know if you have the right size tip, because if it's not the right size, it won't fit. You use the plunger to draw up and dispense the liquid, but wait, there's a trick to it. When you first push down the plunger, it goes all the way to the first stop. But if you push harder, you can push it down to the second stop. The second stop is there because when you dispense the liquid, if you only push down to the first stop, a tiny bit of liquid stays in the tip. Pushing down to the second stop squirts the rest of the liquid out, so you are sure that you transfer all the liquid in the tip. The second stop is used only for dispensing liquid, which means never use the second stop to draw up liquid. To draw up liquid, push the plunger to the first stop, insert the tip into the liquid, then slowly release the plunger. To dispense the liquid, push the plunger to the second stop. Now, in order to draw up liquid, you have to push the plunger down first. But if you do that when the tip is in the liquid, you'll blow bubbles. So always push the plunger before putting the tip in the liquid. When you're drawing up or dispensing the liquid, if you push the plunger button too hard, you can squirt your liquid all over the place. So push the plunger button slowly. Once you transfer the liquid, your tip is dirty. If you use this tip to transfer a second liquid, you'll contaminate the second liquid. To get rid of the tip, use the ejection button, which lets you easily eject the tip. Make sure you eject it into an appropriate container. That's it. Now you're ready to use a micropipette. And maybe one day you'll be the one publishing full research in a journal like Nature.
Okay, so let's get back to what we are going to do during our first week. We'll send you the link of that video. Please go watch it again. It's important that you understand how to use a micropipette for the first week. There'll also be some time on Monday morning during the practical week for you to practice your pipetting skills. So don't be too worried. Just understand how it works. So getting back to what we will be doing in our first practical week. Like I said, we want to get from the Grootnoem leaves or the plant cells to an actual stretch of DNA. So we're going to start by extracting DNA on Monday. This will be a process of elimination. If we look at a plant cell, we ultimately want to get to the DNA that is safely secured within the nucleus of the cell. Think of the plant cell as a treasure chest with a strong outer castle wall. The plant cell wall, and this protects this treasure chest, which is the nucleus containing the precious treasure, which is our DNA. So in order for us to get to our treasure, we have to follow the following easy steps. So first, we are going to smash open the castle wall, also known as the cell wall, and disrupt the cell membrane. This we will do through manual grinding using liquid nitrogen. So the ice-cold liquid nitrogen, the cold temperature will deactivate any of the cellular enzymes and prevent any shearing or damaging of the DNA. Then we're going to wash out the gunk. This includes things like chloroplasts, vacuoles, and mitochondria within the cytoplasm with the sorbital wash. So the remaining cell wall debris will be degraded by reagent 1. And now we finally break open the treasure chest, the nucleus wall, and expose the DNA, our treasure. Think of it as a very precious crystal ball that should be handled very carefully to preserve those long stretches of DNA. So now, after that, we will separate the DNA from the remaining cell debris. The DNA will remain protected in the water, while the cell debris, like proteins and lipids, will dissolve in the chlorophyll. Then we will add isopropanol, in which the DNA is less soluble, so it comes out of solution. And during this step, we will actually be able to see our long, precious strands of DNA. After that, we'll clean it up and dissolve it in water. So after our DNA extraction, we need to check the success of our extraction. This we will do on Tuesday morning, and it's called quality control, or in short, QC. So this entails checking the quality of our DNA. In other words, how clean is our DNA? We will do this with a little instrument called the nanodrop, and you will hear all about how it works during that first practical week. We also need to check the quantity of the DNA. In other words, how much DNA do we actually extract? Is it enough? And this we will do with a little instrument called a qubit fluorimeter. When we have our extracted, cleaned up, and QC DNA, we need to prepare a genomic DNA library. So most of you might ask, what is a genomic DNA library? So imagine the genome like a big book with lots of pages, and each page represents a strand of extracted DNA with its special code that tells the plant how to grow, how to work, and how to be a Grootnoem plant. So now we want to read this book to understand how the Grootnoem plant works. But instead of reading the whole book at once, we are going to read it one page at a time. So the DNA genomic library is like a magical way of getting these pages ready for reading. So how do we get our pages ready for reading? So firstly, we do this by preparing any tears in the pages or nicks in the DNA strands. So during our DNA extraction process, we try to preserve the long strands as far as possible, but it still breaks and shears during this process. And that's what we are going to repair. Secondly, we are going to add an adapter. Think of it as a post-it to each page. And this adapter or post-it will be added to each DNA strand. So this is what it will actually look like in terms of the DNA strands and not books. So there are our DNA strands after it's been extracted. You can see a few nicks in the ends. Then we'll do an end prep, repair, and repair it. You can see no more nicks. After that, we'll add our post-it or our adapter to each end of the DNA strand. You can see it in green. And then from Wednesday to Friday morning, we are going to do the actual sequencing. So as you remember, Alphabet showed you this little machine, the Oxford Nanopore P2 sequencing instrument, P2 solo sequencing instrument. And this is the flow cell that slots into the P2. Let me just show you its flow cell. The actual flow cell is this big. So together, this will actually read, together with this little, the flow cell and the sequencing instrument, it will actually read the pages or stretches of DNA. And that will get us from a stretch of DNA to an actual DNA sequence. So how exactly this little instrument works will be explained by my colleague Larissa, which brings me to the end of my talk. So good luck, and we look forward to meeting you all in a few weeks' time.
All right. Hi, everyone. Thanks, Helmin. I'm Larissa. I'm going to be talking to you a little bit about the bioinformatics or the data analysis. So this is something that we're going to talk a bit more about on Friday when you're here, and you're going to delve into a lot more depth with the data in your second week later on this year. So after you've extracted your DNA, you're going to perform the sequencing. And the thing to bear in mind is that there are different sequencing technologies, and each one might work slightly differently, but the general principles and goals are the same. Okay, and again, I'm going to share the link to this on the chat later on as well. So we're using Oxford Technologies. Your device is going to look different to this one, but it will have a flow cell where you'll load your DNA sample. And every long strand or fragment of DNA has these attachments, the adapter sequence that Almina was talking about, and a motor protein. So you'll load your DNA sample into the flow cell. It contains thousands of these membrane wells, and the DNA will go into one of these wells. And each of them has embedded in it a nanopore, so a pore made of protein. The DNA will tether nearby it, and you'll see it starts unwinding. So it's double-stranded DNA, only one strand goes through the pore. And their purpose is to feed this strand through the membrane at a very controlled speed. Now, the thing to know is that there's an electric current flowing through this pore. And DNA, it's made up of these chemical groups. And as they flow through this pore, there's a very distinct change that takes place, or disruption in the ionic current, and we can measure that. There's little sensors in every well picking up these changes, and it's connected to a computer chip. All right, and from that we can get a sequence strand of DNA, which is what we're aiming for. Okay, so there's thousands of these wells, pore in each of these wells, and one pore can have many strands going through it. Okay, so it's a very popular technology that's really, taken off. And we'll send you the link to this video shortly. All right. So just to know what is going to come off the sequencing machine, once you finish sequencing, what are the files that you're going to have on your computer? We were talking about the signal disruption, or the current disruption in the form of a signal. If you come across these POD5 files, just know that that is what is contained in those files. It's the raw signal. But that's not very useful to us. We can't do much with that yet. We need to interpret that signal and try and understand, well, how does that little squiggly graph tell us what the ATGC sequence of the DNA is. And that's a process called base calling. And it's actually quite a challenge to interpret those signals, but they're getting more and more accurate. And what you will then get is called a FASTQ file. Now that's the file type that you're going to be working with mostly in your week 2. And it'll be the output, the DNA output, along with a vote of confidence. So the base calling software, the software with the algorithm that interprets these signals from the graph, it gives a vote of confidence. How sure is it that it interpreted that signal correctly? That's something that we're also going to talk about a bit later, but just something to keep in the back of your mind. Okay, so what are these FASTQ files going to look like? We've gone from our double-stranded DNA. One strand has gone through the pore, the nanopore. We only need one strand because, as Elsabeth was saying, the DNA is complementary. So if we know one strand, we can infer the other strand. And that strand, we call it a read, and you can think of them as strings. And it'll quite literally be a text file, right? So this FASTQ file, this .fastq extension, it's a file of text. All right, and there's a specific structure to this file and some more to it that we're also going to talk about on Friday, but this is really the core of the file. It'll be the DNA sequence, the ACGTs, for every read that went through a pore with a unique identifier. Now also bear in mind that there were many reads that went through the pore and many pores. So you can have many reads in one FASTQ file and then also many FASTQ files. So it depends on the size of your species as well and how successful your sequencing run was, but you could maybe have 7 million of these reads, all with varying lengths, but within a range and various quality, right? Yeah, so this is our FASTQ format. And these are the reads that you'll then try and find the overlaps, as Elsabeth was also explaining, for your draft assembly later on. Okay. So how much data are we talking here? One of those machines that Helmin showed you just now with one of those flow cells, you kind of plug it in like a USB. One of those can generate about 1 terabyte of data. Okay, so I think you're all very familiar with gigabytes. So there's about 1 billion bytes in a gigabyte. For those of you who are doing computer science, you may know that's not exact, but approximately. And there's 1,000 gigabytes and a terabyte. Okay, so again, this is a really, it's a bit of a hypothetical value because, again, it depends how well your extraction went, how well your sequencing went. And also, it's not just raw DNA that you're getting off that instrument, it's also other information and other files and yeah, so not just raw DNA, but let's say hypothetically you reach that 2 terabyte limit. How much data would that be? Now, bear in mind, this is text data. And it's approximately 5.5 million books, average size books. So that's a lot of data. And how do we deal with all of this data? Well, that kind of brings us to what Renee was telling us right in the beginning. Okay, brace yourself for a few acronyms here. The Government Department of Science and Innovation, they give funding to a body called NICIs. Okay, and that stands for the National Integrated Cyber Infrastructure System. And they do what their name says. They provide computer resources or infrastructure on a nationwide scale, and these systems are integrated with each other. So they're compatible. And there's really 3 main branches at the moment. The first is CenGen, so that's when you want to transfer data. And the other 2 are the ones that we are using at the moment. It's the CHPC and the RISA. All right, so the CHPC, it's the Center for High Performance Computing. And I'm not sure if you've come across high performance computers before, but just as an example, say you were to generate all of this text data that we spoke of. Let's, for instance, say it is 2 terabytes of data. That's a lot of data. You know, your average computer, the laptop that you might be using right now on this call, let's say, for instance, it's about 250 gigabyte storage, all right? And your computing power, if you've got like 8 cores and maybe 32 gigabytes of RAM, that's a pretty good computer. But it's nowhere near what you need to analyze a genome and to deal with this size of data, right? So an HPC or a high performance computer, it's kind of like aggregating all of these computer resources together and giving you access to a lot more resources than you ever would have on your own personal local machine. And so you can run the sort of processing or analysis on your data. And then DRISA, that's the data storage platform. That stands for the Data Intensive Research Initiative. Okay, so that's just a place to store your data long term, and it's a thing that you have to consider because, okay, say you were to buy a whole bunch of external hard drives and put your data on that, well, it might work, but it's not very secure, and you would need to back it up because if something happens to those external drives, all your data's gone. So it's also quite expensive to try and set up your own storage solution. So if you're doing academic research, you can request access to these resources, and they are freely available and really helpful for this kind of processing. So I hope that's given you a bit of a taste of what you can expect at the end of your first week in terms of the data. And we're going to talk a lot more about this on Friday, and we're looking forward to seeing you all.
Thanks a lot, Elmin and Larisha. It might look a bit overwhelming, but we will go through everything slowly again. In terms of logistics, I just want to say that Elmin mentioned that you will work with minus 80-degree cold samples. So we just request, we don't have a specific dress code, but for the first day, please come with closed shoes, so that if there's liquid nitrogen spilled over from your bench on the floor, that you don't get hurt. So that's just for the first day, that's important. Sorry for the sound of that micropipetting video. I want to put a challenge to you, see whether you can find us a better video while you look into micropipetting, and come and show us when you come and visit in March. Yeah, the rest of the logistics we will communicate via our email or WhatsApp group. We have now time for questions to any of the speakers. And we can also say my email address is cengen@cengen.co.za. If there's maybe a journalist or someone that wants to ask more specific questions, you're welcome to write me an email. But the floor is open for questions. Maybe that will be good. Thank you, Elmin. That will be good if you raise your hand, and then I will nominate people to start speaking. Thank you. Are you feeling a bit overwhelmed? No, we will explain everything slowly again and take you through. And you saw it's a big team of trainers that will work with you. So you can just relax, and we will work closely with you every day. No questions from the public or from the education department? Lucy, maybe something from your side?
Hi, Renee.
You can switch on your camera if you want to.
Okay, I don't want to because I'll tell you later why not. But I would just like to, just to emphasize that we're here to learn. And I know that the participants might be a bit shy still. We haven't met face to face. And maybe, yes, a little bit overwhelmed because of all the information, and lots of the information is new to them. I hope that has tickled your brain cells, want to go and find out a little bit more about handling a pipette, about the technology that you're going to be using. And maybe even, I know that in grade 10, all you learn about DNA, you don't necessarily learn about the sequencing, and so that might be also a little bit overwhelming, but I want to tell you that for next year you're just streets ahead of all your classmates when it comes to DNA after your experience. So from my side, Renee, I really want to once again just thank the team for their preparation for this afternoon's meeting, for making it really clear and simplified, so that learners get an idea of what they can expect. But I also want to tell you that many learners have come for a workshop where they've just done a small extraction of DNA, and they've really enjoyed it immensely. And you're just going to go much deeper into the extraction than what they've had the opportunity to. So welcome to this amazing project. And yes, we look forward to meeting you, and don't ever be shy to put up your hand or to ask, because the team is very open to leading you into learning. So thank you so much, Renee.
Thank you, Lucy. Okay, so then we can proceed, I think. Elizabeth has a nice, fun activity which will take less than 5 minutes. So she prepared you for that, but just follow her instructions. Thank you for everyone attending. We really appreciate it. And I see you in March.
Okay, so we're going to do the Mentimeter exercise. I hope all of you have been able to log on to, or to open it on your browser. Don't know if you can see on my phone. No, you won't be able to see, but I hope you've managed to get there. So the code that you need to type in is 41432532, and we're going to go through the presentation together. And there are a few questions that we want to ask you. So remember, it's going to be anonymous. We won't know who answers what. And we would just like to get it sort of an idea of how you think and what you like to learn about and things like that. It's not a lot of questions, won't take a lot of time, but yeah, we're actually just curious. So once you've logged in, you can proceed to the questions, and then we will have a look at the answers together, who's answered most. The first question, what do you like to learn, or most like to learn of in life sciences? It's okay if you don't say plants, we won't be offended. But yeah, we can all let's just have a look. I see humans usually take the lead on this. And we need to get to 13 people for all the learners that are here. I think everyone is here, almost everyone. 7, 10, 11. I know I have to do some maths. I don't do maths often anymore. Okay, so humans, like I said, usually are the things that people like to learn about most. We don't blame you, because in the syllabus, you might think plants are a bit boring. Hopefully we can change your mind when you come. So, okay, humans are the favorite at this point. Next question. Which one do you think would you prefer? Would you prefer to do lab work or computer work? Two types, different types. Coding first, so that's like the really the hardcore computer guys, the scientists. Or do you like to write things? Do you think you would like to write like grant applications and articles or reports on your research? Which part do you think would you like most? It seems like lab work is the favorite. I must admit, I also love lab work. I don't get to do it that often anymore. But yeah, you're going to do plenty of that in the first week. So it's good that you're excited about that. I'm curious to know who the computer persons are. Maybe you can tell me privately if you want to. Okay, next question. Would you eat or drink something that is labeled as GMO? GMO is a highly contended subject. If you don't want to, don't be shy. Like I said, it's anonymous. At this point, it seems like everyone's okay with the GMO, eight people. If you don't know how you feel about it, that's also fine. If you'd like to know a bit more about it, then maybe we can chat to you if you want to. We can just have a conversation while you're here. If you'd like to find out a bit more, just ask. Any one of us can help you or can chat a bit to you and then tell you how it works and what it does. Okay, but at this point, it seems like most of you are comfortable with GMOs and to eat and drink it. In South Africa, we actually do have GMO crops, only 3. It's maize and cotton for clothes, and then also soybeans. So you have probably eaten GMOs before. Okay. What proportion of our genes do we share with bananas? So genes can do lots of different things. As humans, how many of these genes how many of our genes do you think overlap with that of a banana plant? You get different types of genes. Some of them are like we looked at the proteins that do transport. Okay, it seems like the majority says 30%. Well done to the 2 people who said 50%. It seems like half of our genes are similar to those that you find in bananas. You would never think. There was a late one now. Sneaky. Okay, last question. Please write down what you expect to learn and how you feel about this workshop. If you are excited, if you are a bit nervous, how do you feel? What do you think will you learn? What do you look forward to? Anything regarding this workshop, we'd like to know how you feel about this. So if you've written your 2 words on the Mentimeter, you just say submit, and they will come up on our screen that we can all see. Some lab work, rebuilding DNA, building a puzzle. Someone's excited. I'm glad. I think lots of people might be excited, hopefully. Okay. Sequencing, a bit nervous. That's okay. It's totally understandable. Don't worry. We will help you to get through this week. And you will not be judged on your abilities, because you remember you don't know anything yet. You haven't worked in a lab yet, so it's completely okay. Someone's interested. Genetics, DNA. Excited and nervous. Good. Nervous excitement's always good to get you going into, so that you can be receptive for new information. Okay, so it seems like we've got some excited students, we've got some nervous students. Someone cannot wait to learn, start learning, intrigued. Okay, good. Thank you. Thank you for your participation. This is actually the last question. Like I said, we're not going to use this data for anything. We might do some of these questions again at the end. Like we might ask you again at the end of this week, do you still like the lab work? Or do you think the computers would be nicer? So at the end of maybe of the project as well, we once you've written up your report, then we can ask you these questions again, and we'll definitely ask you about plants, humans, or animals again at the end. Thank you for your participation. Thank you for making time for this afternoon and for listening and for being here. We really look forward to meeting you towards the end of March, and I hope you enjoy the rest of the term before then. Thank you, everybody.